था कल और 7th class? 8th class, right? So 8th class, I'll let you know in a minute. Part 6, Part 7, Part 8. On 31st December, at 5 o'clock, on 31st December at 5 o'clock in the evening, the lecture will be uploaded. On 30th December at 5 o'clock, part number 7 will be uploaded. And on 29th December, matlab kal, at 5 o'clock, part number 6 will be uploaded. Okay. But notes though, I have been giving you constantly the notes and all into the group. So that is not a problem at all, right? So shall we start, guys? Yeah. Why only few students today? Remaining, today is Thursday. Sleeping? Remaining students are sleeping. Right. Chalo, let's start now. Today we shall be discussing more clinical part. Clinical part in the sense we shall be discussing about this thing, about Staph aureus. We shall be discussing what are the different kinds of staphylococcal infections we have got. Okay, what is the mechanism and all those along with the pictures and all we shall be discussing. After that, I will also discuss about pneumonia. Okay. I will discuss everything about the pneumonia and yeah, that's all. So all of you now look here. This is the classification which all of you know already, right? So basically we divide this gram-positive bacteria into bacilli, cocci and branching filaments. Every bacteria which we discuss, right? We are starting from this table only. Now, when you look at most cocci are aerobic only because anaerobic cocci, they don't survive basically. Okay. So we discuss the aerobic group itself. Now, this aerobic cocci is divided into two parts. One is called as a catalase positive and the other one is called as a catalase negative. Okay. So one group, you call it as catalase positive. Another group, you call it as catalase negative, cat positive and cat negative. Okay. Now, when it comes to catalase negative, for example, if I'm drawing one bacteria in the form of a chain like this, right? Now tell me these bacteria are circular. If these bacteria are circular, I can call them as cocci and they are arranged in the form of a chain. So I have to call them as anyone? Very good. I have to call them as streptococci. Okay. So I have to call them as streptococci. Any bacteria is in the form of a chain is called as streptococci. So they are called as streptococci. Okay. Next important thing. Am I audible, guys? Am I audible and am I visible to all of you? Right. Next important thing is that there is another group of bacteria. This group of bacteria is in the form of a grapes, a bunch of grapes, right? Bunch of grapes, they don't have a regular pattern, right? Irregularly, they are located like this. Very good, Fatima, very good. So this is called as what? These group of bacteria are called as Staphylococci. Staphylococci. So two groups of bacteria we have got over here. One is called as streptococci, another one is called as staphylococci. Okay. Now, the most important and the exam asking question, definitely this question is going to come in your exam. 100% this question will come. So far, there is no year where they did not repeat this question. Okay. So what is the important question is that this particular staphylococci is divided into two parts. Okay. Some staphylococci release coagulase enzyme. Some staphylococci do not release coagulase enzyme. If you are releasing coagulase enzyme, they are called as coagulase positive staphylococci. Okay, so you call it as COPS, COPS, coagulase positive staphylococci. अगर coagulase enzyme is not released, you call it as cons. Cons मतलब coagulase negative staphylococci. Coagulase positive, coagulase negative. When it comes to coagulase positive, the only example which we have over here is Staphylococcus aureus. Staphylococcus aureus. If you remember, when I was teaching you the pigments that are released by the bacteria, I told you Staphylococcus aureus will form what kind of colonies? If you remember, everyone. Jaldi batao. Very good, Ajoy. Very good, Arbina, Nidhi, Nishoka, very good. So they release what kind of colonies? They release golden yellow colonies. एक चीज याद रखो, Staphylococcus aureus, aureus in the sense gold. Aureus in the Latin term is gold. Okay. When it comes to cons, coagulase negative staphylococci, we divide these cons into two types. What are these two types? One is called as Novobiocin sensitive. Another one is called as Novobiocin resistant. अभी ये Novobiocin का concept क्या है? All of you open your ears and listen carefully. What is Novobiocin is? It is an antibiotic. Similar to Novobiocin, there are other antibiotics. For example, there is another antibiotic called bacitracin. Another antibiotic called optocin. ठीक है? ये लिखने की जरूरत नहीं है. I will write down these things later on when that names will come. Okay. So some of the bacteria are resistant to this antibiotic. Some of the bacteria are sensitive to this antibiotic. How will you know? How will you know is that? ये देखो, I have taken an agar plate. Okay. Now in this agar plate, I have divided the agar plate into 50-50 portions. Okay. Now one side I have added a bacteria called as A. Okay. Let us say that one side I have added this particular bacteria, right? This particular bacteria which I've added over here. ये देखो ये वाला. This particular bacteria which I've added over here. This is called as Staphylococcus epidermidis. Just for an example, I'm telling. Staphylococcus epidermidis. This is the bacteria which is present on your epidermis. On the right hand side, I have added another bacteria called Staphylococcus saprophyticus. Okay, just roughly I'm telling you two examples. Staphylococcus epidermidis, one side I have added like this, I have coated this bacteria, and on the other side I have coated another bacteria called as Staphylococcus saprophyticus. Now what will I do? I will take a tablet. This tablet is called as Novobiocin tablet. Novobiocin, what is it? It is an antibiotic, I told you. I will put a tablet on the right side. I will put a tablet on the left side. You can see in the picture that blue color tablet which I kept, one on the right, one on the left. What antibiotic will do? It will kill down the bacteria. Yes or no? If I put antibiotic here, it will kill down the bacteria surrounding it only. It won't kill down the bacteria which is present far away, right? So you can see where Staphylococcus epidermidis is present. There surrounding the antibiotic, you see there is all the bacteria surrounding that is dead. That is why you can see you can say that you can see a pale zone, or you can see an empty zone surrounding this tablet, whatever I've kept inside. So can I tell you like this, that this tablet has successfully killed all the bacteria surrounding it, forming what? Forming a zone of inhibition. This is called as zone of inhibition. So look on the right hand side then. On the right hand side, you have got Staphylococcus saprophyticus. I've kept a tablet there, that is Novobiocin. But surrounding that, do you find any zone of inhibition? It means the tablet does not have a capacity to suppress this bacteria, to kill this bacteria. It means this saprophyticus is resistant to Novobiocin, whereas Staph epidermidis is sensitive to Novobiocin. So here there is no zone of inhibition. All of you understood this? Or am I speaking only to myself? You're all understanding, right? Right. So here, that is the concept here, which is Novobiocin sensitive, which is Novobiocin resistant. From these examples which I have given, what will you pick up? Novobiocin sensitive is epidermidis or saprophyticus? Jaldi batao. Novobiocin sensitive is which one? Epidermidis or saprophyticus? Very good. Novobiocin sensitive is staph epidermidis, and on the other hand, this is staph saprophyticus. Okay, so one is called as your staph epidermidis and the other one is called as your staph saprophyticus. So this is a picture which I have drawn. Now what I will do is that I will show you the real picture and you will tell me the answer. Okay, ye dekho, this is the same procedure over here. I am putting it as A, I am putting it as B. Now, what is B? Which bacteria is B? Batao, what is this bacteria that is quoted here? First of all, what you have? Very good. Syed, Fatima, very good. Nidhi Jain, everyone, very good. Sultan, very good. So this bacteria which is present here is staph epidermidis. Why? Why are you calling it as staph epidermidis? क्योंकि यहाँ पे what is there? You see this? This is called as zone of inhibition. Okay, you see this, that there is a zone of inhibition over here. So this is a zone of inhibition which you can see. Okay, but on the other side there is no zone of inhibition. So I would put this bacteria under this one, staph saprophyticus. Okay, this is staph saprophyticus, and this one over here is staph epidermidis. So till here all of you are clear? Yes or no? Everyone, jaldi fatafat. Very good. Now what I will be discussing, we will discuss about three important bacteria over here. First, we shall be discussing about Staph aureus. Okay. First, our topic of discussion would be about Staph aureus. Uske baad, we will discuss about epidermidis and saprophyticus. Now, from this, definitely one question will come. I told you, right? That question is from Staph aureus. 100%, either in microbiology or in orthopedics, right? Definitely, or in dermatology. From these three subjects, definitely one question will come from a topic called Staph aureus. Okay, yaad rakho. So coming to Staphylococcus aureus, I will just write it as Staph aureus. Listen carefully, don't write anything at all. So Staph aureus, you know, by looking at these pigments over here, you can see that it is golden yellow color colonies. The colonies here are golden yellow color colonies. Golden yellow colonies. So these colonies over here are golden yellow colonies. Now, what did I tell you? For example, if you're going for a war, you cannot go nakedly to the war, right? So what you will do, you will take weapons and then go to the war. In the same way, the bacteria, when it enters into our body, it will also bring some weapons. These weapons are called as virulence factors. Okay. What are these weapons called? These are called as virulence factors. So if you look at the virulence factors over here, let us say this is one Staph aureus. You see a chain of Staph aureus are present here, right? Chain todi hai ye. This is in the form of grapes irregularly, right? So irregularly I have to draw, right? Now on the surface of Staph aureus, there are some structures like this. You see, there are some spike-like structures. These spike-like structures which are located on the Staph aureus is one of the very important virulence factor, and the name of this is protein A. What is the name of this virulence factor over here? The name of this particular virulence factor is protein A. What does this protein A will do? Whenever a macrophage is coming and attacking the bacteria, this protein A is having spikes all over. This bacteria is having protein A spikes. So what will happen? It will not let any phagocyte or any macrophage to come and eat it up. So what it is exactly doing? What it is exactly doing is it prevents phagocytosis. It prevents phagocytosis. It prevents what? It prevents phagocytosis. Very, very, very important. So this is what? This is anti-phagocytic. This is anti-phagocytic. This is the only point you need to remember. But what comes in the exam, that is very important. What comes in the exam is that you have to know what toxins that are released by Staph aureus. You know, previously we discussed toxins like cerulite toxin, tetanospasmin, tetanolysin, right? All these different types of toxins. But the single bacteria itself will release many toxins. Let us discuss what are the toxins that are released by these Staph aureus. Okay, let us see here. You can just remember by the mnemonic REST. REST, rest, but don't rest now. So all of you look here. The first important mnemonic over here, R stands for rapid onset of food poisoning. Rapid onset of food poisoning in the sense the moment I eat the food, I will get food poisoning immediately. So it means, can I tell that if I take such toxin, the symptoms will be very fast. Why the symptoms are very fast? Because incubation period is very less. Immediately after 2 hours after eating the food, you will start getting the symptoms. That is why it is called as rapid onset of food poisoning. Rapid onset of food poisoning. Now, regarding this rapid onset of food poisoning, what is the toxin that is causing rapid onset of food poisoning? Very good. Very good, Saida. Very good. It is enterotoxin. It is enterotoxin that is causing rapid onset of food poisoning. Second important one, what is the mnemonic? E. E stands for what? Exfoliative toxin. Exfoliative toxin. What do you mean by exfoliation? Exfoliation means the skin, layers and layers of skin. Sometimes, you know, in the winter season and all, you see the skin that is exfoliating from your hands. That is called exfoliation, right? Third important thing. What is the third important thing? S. S stands for what? S stands for staphylococcal skin scalded syndrome. What does this mean? Staphylococcal skin scalded syndrome. So normally in the books it is also given as SSSS. Okay, staphylococcal skin scalded syndrome. So in staphylococcal skin scalded syndrome, what is happening? Skin is going away in the form of layers. Why it is going in the form of a layer? Because it is going to release in staphylococcal skin scalded syndrome, a toxin is released. वो toxin का नाम क्या है? What is the name of the toxin? Exfoliative toxin. Okay, that would cause this particular thing here. You see layers and layers of the skin. The complete epidermis is going away, right? So this is called exfoliation. Whenever you see this picture, if they ask you what toxin, what will you tell? You have to tell it as exfoliative toxin. If they ask you what is this condition, you have to tell SSSS, staphylococcal skin scalded syndrome. Clear, all of you? Fourth important thing is last. What is this? T. T stands for what? Toxic shock syndrome. Very, very important. ठीक है, टॉक्सिक शॉक सिंड्रोम में टॉक्सिन का नाम तो बताओ. व्हाट इज द नेम ऑफ द टॉक्सिन हियर? टॉक्सिक शॉक सिंड्रोम टॉक्सिन नंबर 1, TSST1, toxic shock syndrome toxin number 1. So these are the 4 important things that are released here. Out of this, यहाँ पे toxins कितने discuss किए हमने? One is called as your enterotoxin, one is called as your exfoliative toxin, one is called as your TSST1. So all of you understood this? Yes or no? जल्दी बताओ. Very good. Only Nidhi understood. बाकी सब चाय पी रहे. Very good. So now here, what is another important concept is, I will talk about these toxins in a minute, but what is the important concept is, what are super antigens? Antigen होता है एक, super antigen भी होगा. What are the super antigens here? There are 2 different types of super antigens in this topic. One is called as TSST1, जो हमने अभी डिस्कस किया. This is called as super antigen. Another one is called as SSSS toxin. मतलब स्टेफाइलोकोकल स्किन स्काल्डेड सिंड्रोम कॉजिंग टॉक्सिन, उसका नाम क्या है? एक्सफोलिएटिव टॉक्सिन. सो दैट इज नथिंग बट योर एक्सफोलिएटिव टॉक्सिन. एक्सफोलिएटिव टॉक्सिन. So these are the 2 important toxins which are called as super antigens. These toxins here are called as what? These toxins here are called as superantigens. Is everyone clear? Now, when it comes to exfoliative toxin, there are two different types of exfoliative toxin. One is called as exfoliative toxin A, exfoliative toxin B. Okay? So overall, if you see here, TSST1, exfoliative toxin A, exfoliative toxin B, these three are called as what? Superantigens. Now let us see the concept of superantigens. So by the way, tell me what are superantigens? जो आप जानते हो बताओ. Superantigens, Superman. What are superantigens? Antigens which work superly are called superantigens. Very good, Nagesh Paila. Very good. All of you, please subscribe to Nagesh Paila YouTube channel also. He teaches biochemistry there. Antigen which resembles self-antigen, no. Toxin to bacteria से आ रहा है या फिर ये antigen कैसे? Sultan, see, any foreign substance that is entering into your body is called antigen. Any foreign substance. Poison is also within the poison. Let us say snake bite. There is poison in that. Within that poison also there are antigens. These are called antigens which will destroy your body. Okay. Anything, the outer part, anything which is entering into your body from an organism, that is called as an antigen. Okay. Right. Now all of you look here. What is a superantigen is that, see, this concept is really very important. Just understand this. Your pathology will be easy. All of you just put your pens down. Just look here, whatever I'm drawing. Everyone, okay? See, let us say this particular cell which I'm drawing right now. This particular cell which I'm drawing right now is called as a T cell. What is this? This is called as T cell receptor or T cell. This is simply called as T cell receptor. Okay. So I will draw. Okay. Leave it. This is called as what? T cell. ये देखो, ये है T cell. ये अरे this is T cell or T cell का receptor है T cell receptor. Everyone understood? Everyone now listen carefully. This is called TCR, and this is T cell. TCR मतलब T cell receptor. Now T cell receptor is having two important parts. One is called as alpha domain, another one is called as beta domain, or alpha subunit or beta subunit. जैसे अच्छा लगता है वैसे बुलाओ. Okay. Now what I'm doing is I am drawing a macrophage here. This is the macrophage over here. Okay. Now on the surface of macrophage, there is another receptor in this way. This receptor is called as MHC class 2 molecule, major histocompatibility class 2 molecule, MHC class 2 molecule. So basically what happens is, let us say a bacteria entered into my body. Abhi normally macrophages kya karega? Macrophages, this is bacteria. Macrophages will go like this. It is going. Acha, bacteria hai? Okay, fine. So by a process called as endocytosis, this macrophage will completely engulf this bacteria. So I am macrophage, I have completely taken the bacteria inside me. और जैसे bacteria अंदर जाएगा, within the macrophage, what will happen? The bacteria is broken down into pieces. Okay. Out of all, let us say bacteria is broken down into 20 to 30 pieces. Out of this 20 to 30 pieces, this macrophage will vomit one piece like this, one piece, and that piece will come out. And now I will take this piece on my hand. Okay. So exactly this is the piece which I'm talking about. Yeah, देखो. This is the exact piece which I'm talking about. Okay. So this is what, this is an antigen only. When you're cutting down the every part, every inch in the bacteria is called as an antigen. So you cut this, and one part you're taking it outside. And what you do is that you go, you wander everywhere, and finally you go fix into this. You go and fix into that T cell receptor. Now here, one more important thing I want to tell you, just keep this concept aside. One more important thing I want to tell you is that, ये देखो, ये, this is a another different type of T cell receptor. Okay, let us say this is T cell receptor number 2 और ये वाला T cell receptor number 1, इसका नाम देते हैं 1 2. Okay, now I am talking about another different type of T cell receptor. ऐसे होगा. Okay, let us say that this is T cell receptor number 3. Now you will only tell me the answer that this particular MHC class 2 molecule, this particular MHC class 2 molecule is searching for a perfect T cell. It is searching for a very perfect T cell. Which T cell it will go and fix? बताओ कहाँ पे जाएगा? See, will it go and fix into this? Can it fix here? You see, antigen is circular. Antigen is circular, but can it go and fix into a triangular depression? No. Next, will it go and fix into this rectangular depression? No. It can go and fix only to this particular T cell, that is T cell receptor 1. So it means T cell receptor 2, type 2 T cells, type 3 T cells, they are not activated. Same concept like enzymes. Janvi, very good. So type 2 and type 3 are not activated, only type 1 T cells are activated. Okay, okay, activate ho gaya. Keep this thing aside. Now what will happen is that T cell is so happy. Now what T cell will do? T cell should thank macrophage. अच्छा हुआ कि तुम लेके आया मेरे पास, है ना? So this T cell, how will it thank is that this T cell will throw a chemical. ये chemical का नाम है interferon alpha, interferon gamma, IFN gamma, which means interferon gamma. So it will throw interferon gamma onto the macrophage. Now macrophage is so happy that it received a credit called as interferon gamma. Now in that happiness, what macrophage will do is that macrophage will release 3 important things. Macrophage will release how many important things? 3 important things. What are those 3 important things? One is called interleukin 1. Another one is called as interleukin 6. Another one is called as TNF alpha. Interleukin 1, interleukin 6, tumor necrosis factor alpha. These are the 3 things that are released by macrophages. Okay? Out of what are the individually in local functions alag alag? I will tell you that. Okay, but overall I'm discussing the concept. Overall, all of these 3, you know what they do? First important thing, they cause fever. Second important thing, they cause hypotension. Blood pressure niche girega. Third important thing, they will cause desquamation. Desquamation. Desquamation matlab, desquamation is same like exfoliation only. Okay, desquamation. So this is the thing that is happening over here. Normally, this is the thing that happens in you and me also. Till here, is everyone clear? Perfectly clear? I did not still teach you what is superantigen. Not 6, only interleukin 1, Raj, only interleukin 1. You understood this, right? Everyone understood this? If you give me a, yeah, very good. So now look here. Out of all this, only one group of T cells are activated. This would lead to desquamation. This is called desquamation of skin. Okay, this is called what? Desquamation of skin. अभी ये desquamation जो है painful होगा कि painless होगा बताओ? How will the desquamation? Just look at the picture and see how red it is. Yes or no? Don't you think patient will have very painful desquamation? Yes or no? Wrong. Sultan is right. This is painless desquamation. This kind of desquamation is painless desquamation. Okay? Completely painless desquamation. Okay? Everyone is clear with this now? Right. Now look here. Let us discuss about another important thing. So 3 types of receptors I told you, right? एक ये वाला था. This was the receptor. Sorry. This was one receptor we had. This was another receptor we had. This was a third receptor we had. Okay. So all of you look here. This is 1. This is 2. This is 3. Okay. 1, 2, 3. Now if I will draw one more important thing and you will tell me the answer. Okay. So let us say that this is MHC class 2 molecule. Okay, MHC class 2 molecule here. This MHC class 2 molecule is carrying an antigen like this. Okay. Now tell me this particular macrophage with MHC class 2 molecule, where will it go and fit? 1, 2, 3. Batao. Where will it go and fit? Very good. It will go and fit into 2. So do you think it will activate 1 and 3? It won't activate 1 and 3. What is super antigen now? Abhi ye dekho what is super antigen. Now here the topic is little bit tricky. This is your macrophage. Macrophage ke upar hoga. What is this? This is your MHC class 2 molecule. और ये आ गया super antigen. ये देखो, this is your super antigen. You know what super antigen will do? Super antigen को ना receptor से कोई मतलब नहीं है. It has to nothing to do with the receptor. It doesn't care the receptor at all. What super antigen will do is that instead of going and attacking the center here, this super antigen will go and activate this T cell receptor from the side like this. Okay. Now another one will go and activate from the side. Another one will go and activate from the side. When it is activating from the side, do you think is receptor playing any role here? Shape of the receptor is playing any role? It is not playing any role. So what does this mean? It means that all the T cell receptors, T cell receptor number 1, T cell receptor number 2, T cell receptor number 3, all of them are activated. एक बार ये सोचो कि if all these receptors are activated, then these receptors in turn activate your macrophages with the help of interferon gamma. I told you interferon gamma, right? अब क्या होगा? This macrophage will release interleukin 1, 6, TNF alpha. This macrophage will release interleukin 1, 6, TNF alpha. This macrophage also will release the same. It means patient is having fever here, here also, here also. Patient is having hypotension here also and here also. Patient is having desquamation here and here. It means what I'm trying to tell you is that pehle only one group of T cells used to be activated. Now in your body, let us say there are 8 groups or 10 groups of T cells, all of them will be activated. When all of them will be activated, there is massive activation of your immune system. Okay, massive activation of your immune system ki wajah se there is massive release of these 3 important cytokines: interleukin 1, interleukin 6 and TNF alpha. So tell me, in this condition patient was having fever, but in this condition patient is having severe fever, very high fever. Okay, next, in this condition patient is having hypotension, but in this hypotension, which means blood pressure from 120 by 80, it dropped down to 100. Okay. Now here, because of severe hypotension, blood pressure from 120 dropped down to 70. So sudden drop of blood pressure is called as shock. Sudden drop of blood pressure is called as shock. Okay. Third important thing, here patient had desquamation, but here the patient will have very severe desquamation. This entire thing is a concept of super antigen. All of you understood? Yes or no? Jaldi batao. All of you understood, right? Then if you are all understanding, why few students? What about the remaining students? मुझे भी इंटरेस्ट होना चाहिए क्लास बताने के लिए. You see, AC remote is also not listening.
Sorry, there's something wrong. Please try again.
Everyone's chat is off. 9. It is on. I can look at your chat. I can see. Then how you guys are chatting? How you guys are sending messages to me? I off the option. private messages. ये क्या है private message? We can't see. Yeah, you can only message to the host. I am the host. Give me a second. I'll sort out this. Chalo, they might be busy. They're not lifting the call. But when I get a call back, I'll sort it out. Okay. Right. So this is a thing over here. In AIMS exam, you know what question they have asked? Hello. Right, guys. So the problem will be solved now. All of you can look at the chat. So all of you look here now. So this is the thing. Now you tell me now, by looking at this picture, here there is alpha, here there is beta, alpha subunit, beta subunit, alpha subunit, beta subunit, alpha and beta. Now tell me, where is the super antigen attaching to? Alpha